Assalamualaikum warahmatullahi wabarakatuh. Welcome to the Biotechnology of Plant Protection class. After the mid-semester exam, my name is Miftahul Ajri. And for the first week, we will still discuss its relation to the weeds, yeah. And then we will discuss also about the plant disease biotechnology. So today we are going to discuss about herbicide plant resistance. Okay, so the learning outcome, students are able to explain the use of herbicide resistant plants or how herbicide resistant plants are utilized. Okay, so biotechnology is a field within technology. And like all technologies, it must stem from a well-defined problem, yeah, and also serve as a solution for increased efficiency. You may have heard about countries that growing rice indoors or in a building. I think that is remarkable technology. But the question is why haven't we adopted it here in Indonesia? Why haven't we adopted? Or perhaps uh, we have wondered why some countries have advanced agricultural technologies that we don't yet use. Yeah, I think it may be a question for us. Yeah, because technology needs to address relevant issues. In Japan, for example, only parts of the country in the southern regions have a tropical climate that's suitable for year-round rice cultivation. And these areas are much smaller than subtropical areas. So they developed indoor rice cultivation. But here in our country, in our tropical climate, we can grow rice throughout the year without such problems. So the technology must be effective, yeah, fulfilling its purpose. Maybe it might not be needed today, yeah, but who knows for the future we may require. Okay, starting from the root of the problem, we identify possible solutions, design, and plan the technology. The solution may not always require advanced biotechnology. Sometimes a simple technology, maybe a proper cultivation methods or irrigation schedule, maybe. Yeah. So simplicity is often adequate. Yeah. And complex solutions are not always necessary. Beside that, yeah, we have to also assess and also manage the risk, considering if the technology poses dangers to humans or the livestock or ecosystem. You can see at the picture, yeah, we can see a drone that's spraying herbicides on crops. And the spray may affect what target organism, yeah. The target organisms, of course, are weeds, yeah. But also, the spray can affect non-target organism, yeah, which is cultivated plants. So pesticide spray that misses the target organism is known as drift, yeah. As I write here, herbicide drive spray. Yeah. So herbicide drive spray or drift. I mean, sorry, or not drive, drift. Yeah. Herbicide drift spray is a pesticide spray that miss the target organism. Yeah. Okay. So as we know, yeah, the crop like corn, soybeans, uh, the crop like corn and soybeans face weeds problems. Yeah, so many weeds that grow in the cultivation. And applying herbicides before crop emergence doesn't pose issues. Yeah, but uh, how when we apply herbicides after the crop emergence, yeah, after the crop has been cultivated. Yeah, so once crops are growing, herbicide application becomes challenging, of course. So one herbicide company, yeah, also they produce seeds with a technology named herbicide plant resistant, yeah, herbicide resistant hybrid, yeah, crop varieties. So the herbicide, something like in the picture, yeah, can be sprayed uniformly across the field, but still ensuring crop safety. Yeah. So the herbicide just affect for the weeds, yeah, but not affect for the cultivation, yeah, soybean, cotton, or corn. Okay, and this is the technology name herbicide plant resistance. Yeah. So the next question is how herbicide resistant plants are created. Okay. Um, we have here one of the herbicide types. Yeah. Glyphosate. Yeah. So glyphosate is a widely used herbicide. Yeah. Very widely used. Yeah. It's very common to use glyphosate in a weed control. Yeah. Uh, one one such herbicide resistant plant is resistant to glyphosate. Yeah. And the glyphosate targeting an enzyme in the shikimate pathway called 5-enolpyruvylshikimate shikimate triphosphate, or we can say EPSP synthase. Yeah. So actually, this pathway, yeah, this shikimate pathway exists in plants, in fungi, and some microorganisms, but not in animals, yeah, including humans. So it's making glyphosate relatively safe for animals and also the humans. But however, yeah, it is deadly to plants relying on this pathway. Yeah. So the EPSP synthase enzyme is vital for producing essential amino acids, yeah, aromatic amino acids. And we know that the aromatic amino acid form the protein and required for normal growth. Yeah, so the aromatic amino acid going to protein synthesis, and then it's affect about the growing of the plant. And glyphosate binds to EPSP synthase, yeah, blocking the production of these amino acids, yeah, and of course, killing the plant. Okay. And then herbicide-resistant plants carry the gene coding for a glyphosate, yeah, inserting a gene encoding EPSP synthase, yeah, that is insensitive to glyphosate. Yeah, it is originating or obtained from Agrobacterium sp strain CP4. So these transgenic plants, yeah, the herbicide resistance plants for the glyphosate, um, have EPSP synthase, yeah, which continues to function despite glyphosate application, unlike ordinary plants where this enzyme is inhibited. Yeah, so EPSP is still working in the herbicide-resistant plants. Yeah, okay. It's originating from Agrobacterium strain CP4. Yeah, so the name of the EPSP enzyme is CP4 EPSP. Okay. Okay, since the introduction of glyphosate-based herbicides, there are planted with glyphosate-resistant crop, yeah, that has grown significantly. By 2009, nearly 60 million hectare of soybean, cotton, and also corn that contained a modified APSPS gene, yeah, that confers resistance to glyphosate. Yeah. But however, yeah, with the extensive use of glyphosate, with resistance has also emerged. Yeah, certain weeds like Amaranthus species, yeah, we know that, have developed resistance, posing significant challenges across corn and cotton producing areas in the United States. Okay, and also with resistance to a photosystem 2, PS2, inhibiting herbicide, the name of the herbicide suggests atrazine, has also been documented across many corn growing areas of the USA. Yeah, and also in the other region, Amaranthus tubulatus, yeah, have been identified with resistant to herbicides that inhibit several modes of action of herbicides, yeah, including ALS, PPO, EPSPS, yeah, of course, and also HPPD. Yeah. Okay. So yeah, herbicide resistance in weeds not only complicates with control, yeah, but also raises agricultural costs and reduces crop yields, yeah, of course, yeah. And multi-resistant weeds, yeah, multi-resistant weeds. So they have a resistant to many mode of action, uh, more complex, yeah, and of course costly manage, and of course take more expensive management strategies. Okay, so we talk about the prime strategy for managing herbicide resistant weeds, yeah. Okay, because to make uh, plant resistant, herbicide plant resistant, it takes a lot of time. It takes a lot of effort. Yeah, takes a lot of cost. So we have to make a strategy for managing herbicide resistant, yeah, in weeds. Okay. Um, the key concepts in weed resistant evolution is include selective or selection pressure. Yeah. So the repeated herbicide use, especially on the same crops, will increase selection pressure on weed population. So it can allowing resistant individuals to survive and reproduce, yeah, uh, eventually leading to a dominant resistant weed population. Yeah. So the strategies that can manage herbicide resistance, it can be rotation or mixtures. Yeah. Rotation sequences and mixtures generally have the greatest effect in delaying resistance when the mechanism conferring resistance is target based, yeah, or the weed species are highly self-pollinated, or maybe the seed spread is restricted, yeah. The herbicide mixtures may delay resistance longer than rotation, yeah. But you have to make sure that the herbicide that you mix is make a good combination, yeah, not to be antagonistic combination. Okay, yeah. So uh, for the first time spraying the herbicide, there only one resistant biotype, and then for the next spray, the next time, by the time uh, eventually majority resistant, yeah, or the resistant biota become majority, become dominant. Because the resistant plant can survive, yeah, and set seed. Okay, and uh, it is also necessary, yeah, to distinguish between species shift and also resistant. So in species shift, in a field, initially there are two types of plants, yeah, plant A, and we can see plant B, yeah. Plant A is dominate, yeah, then plant B. So after being sprayed with the herbicide, many plant B are still alive, yeah, while plant A dies, yeah. So that over time the land becomes dominated by plant B. And then while in the resistant process, in a field, there is one species of the same plant. When sprayed with herbicide, there are plants that are able to survive, yeah, this resistant plant. And then these plants make uh, growing, develop, yeah, make grow, grow, and then over time, the land is dominated by the resistant plants. Okay. Okay, well, you can read the following journal to find out more about the herbicide resistant management strategies. In this journal, it states that an effective way is not to spray pesticides often. Yeah, so not to spray herbicides often. Okay, uh, then we go to the gene flow. Yeah, why we discuss about the gene flow? Because the gene flow is suspected to contribute to with resistance. Yeah, so the herbicide resistant plants may transfer the resistant traits, yeah, or gene to the wild weeds. Yeah. Okay, so three types of gene flow, yeah. First one is vertical, and then horizontal, and also diagonal. Horizontal and diagonal, it's quite rare, yeah. But the vertical one, because it occurs between sexually compatible individuals, yeah, such as between cultivated plants and their wild relatives. And then for the horizontal and diagonal, yeah, the horizontal between distant related species, yeah, this is rare. And then for the diagonal between related but not fully compatible species. Okay, so gene flu can happen through pollen, yeah, seed, and also vegetative propagules. Yeah. And then how to prevent this gene flow, yeah? We can do temporal isolation. Yeah. So for the temporal isolation, we synchronize flowering times to prevent cross-pollination. And then the second one, protective vegetation barriers. Yeah, so planting different crops around the main crop to block pollen, maybe, yeah, or seed spread, yeah, to come to the field. And then the biological barriers can do by cytoplasmic male sterility, maternal inheritance, and seed sterility. Okay, so that concludes, yeah, today's material. We'll continue with the next topic. Thank you.